All right, chapter 18. Okay, we're going to lay the groundwork for Wednesday, and before we really get into protein structure, you have to appreciate the basic building blocks of protein, which are amino acids. In the early 1900s, it was believed that protein molecules were the source of the genetic code or genetic information of an organism. Where did they get that thinking? Well, proteins were made up. They knew at that point that there were 20 different amino acids. And they thought, well, if there are 26 letters in our alphabet and we can write 500,000 or more words, different words with 26 letters, what would we be able to do chemically storing information with 20 amino acids? We'd probably be able to write a lot of information, code a lot of information into molecules. So coding in a chemical sense. And in the early 1900s, they knew that DNA existed. They knew that nucleic acids existed. They knew what nucleic acids were in the body, but they did not know what they did. And what has been the legacy of DNA is that what we don't understand, we think is not important. So when we didn't understand DNA, they just brushed it aside and they focused on protein. And then in 1920s, 30s, 40s, it became clear that, hey, we think this molecule is very important. And then in the 50s, it became so important that Watson and Crick got the Nobel Prize for figuring out what the structure of DNA is. And even up to 10 years ago, people did not know they knew what they called the coding portion of DNA is, the portion of DNA that codes for protein. That was it was actually less than 10% of the DNA molecule. The other 90%, they were saying, was the residual of the evolutionary process, that this was, they called it junk DNA. That term is dying right now, actually. If you hear the term junk DNA, you should question it, because it is the legacy of 10 years, 15, 20 years ago. Just 10 years ago, and I'll show you this in chapter 20, an international consortium of biochemists and scientists have come out with a report on the junk DNA portion of DNA and saying, it's actually controlling the other 10%, that it's signaling back to the other portions. So the thought that was non-functional is dying. My textbook that I'm using right now this semester on the biochemistry of cancer uses the term junk DNA. It was written 2013, so 5 years ago. It's still using that term, but it's really not it's a legacy or a obsolete reference at this point in time. Okay, all that to say, proteins are very important, but they're not the hub of the genetic information. What do? They do lots of stuff. Regulation, enzymes, biological catalysts. That's going to be our focus of chapter 19. So enzymes are proteins. They transport. You just saw embedded in the cell membrane certain proteins that allow the transport, or they will actually transport molecules around in the cell, certain protein molecules. Structural, defense, movement, nutrients, they do lots of different things. They're made up of amino acids. Now, this, in January, if I said amino acid, it might not have brought much to mind. But now that I say it, amino should bring to mind something like that. Acid should bring to mind something like that. Put a carbon in between those two. Put a hydrogen up there. And then the only thing we need to really focus on is what is this fourth thing? What's in this fourth position? And this is your alpha carbon. Alpha to the carboxyl group. We've got a carboxyl group here, and that is the next carbon to it. That's the alpha carbon. So these are all alpha amino acids. There are 20 of them. So we got an amino, we've got an acid, we got an alpha carbon, and coming off here is an R. And there are 20 different ones that your body needs to make all the protein. 20 different R groups. All right? So, first off, you should appreciate what is the general structure of an amino acid, an alpha amino acid. Now, notice the difference between what's on this slide and what's on the board right now. What's on this slide, or what's on the board, there's a hydrogen here, and there's only 2 hydrogens here. Well, we've lost a hydrogen at physiological pH. Physiological pH is approximately 7.4, just slightly above neutral. That hydrogen will hydrolyze off of there and form a carboxylate ion, and will reside over here on the nitrogen that has a lone pair, a non-bonded pair of electrons on it. You get an NH3 plus, a COO minus on the same molecule. So what does that look like? That's German term, Zwitter ionic. It's carrying a positive and negative charge simultaneously. One end has got a positive charge, the other end has a negative charge. Zwitter ion. Here's the non-ionic form. Can it be in that form? Well, at a lower pH, yes. It would be in that form. But at physiological pH, it would become the Zwitter ion. Okay. I'm mentioning there's 20 different R's. We're going to break those into 5 different categories, 5 different classes. When I hand you the homework here at the end, you'll see that there is 1 amino acid in each one of these categories that I expect you to memorize the structure of. All right, so I don't expect you to memorize all 20 of these. I should tell you the good news first. I told you the bad news first. Good news, you don't have to memorize these 20 amino acids, but I do want you to pick out, and I'll show you which ones. I'm picking out one out of each category so that you can remember some of these. And I think, all right, so well, let's just see what the 5 categories are. They're going to be categorized according to the nature of the R group. What is this R group like? Is it nonpolar? Is it polar but uncharged? Aromatic? Positively charged? This would also be, I mean, this is polar, water loving, but it goes much further. It's even positively charged. it's going to have an amine group on it that has not this amine. There's going to be another amine on the R that can pick up a proton and be positively charged. There's going to be a carboxyl on a couple of them, not this one. In addition to that one that's on every amino acid, there'll be another one, and it can lose that hydrogen and become negatively charged. So now let's look at this category. The next slide is looking at this first category here. Here is an isopropyl group. Remember back in chapter 10, alkanes are nonpolar, right? They're hydrophobic. They're not soluble in water. These molecules are not water loving. They're hydrophobic. Now notice that carbon. How many different things are on that carbon?
4.
Four different things. Hydrogen, carboxyl, an amine, and a methyl. When you have 4 different things on a molecule or on a carbon, what kind of carbon is that? What? Chiral carbon. So when you have a chiral carbon, you can have a set of enantiomers. You can have an isomer. So there is going to be an L and a D isomer. And if you went along and looked at all of these, 4 different things. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. These are all chiral carbons, except which one?
The first one.
Glycine, the first one. 1, 2, 3. I can't say 4 because those are 2 the same. So it has 3 different things on it. Glycine is the only one that is not optically active. The other 19 come in an L or D variety. And L is the biologically active isomer of the amino acids. L. So saccharides, it's D. Amino acids, it's L. We'll mention that again in a minute or 2 here. There's a certain amount I want to try to get you to so that we're all set up for the activity on Wednesday. Okay, polar but uncharged. Now you're at the point in this course where you should be seeing, when you see a hydroxyl group, you say, wow, that's kind of like an alcohol. And alcohols, an OH bond is a polar bond, right? So that bond is making this R group have a polar or water loving, a hydrophilic nature to it. These are polar but uncharged. Polar but uncharged. You've got nitrogens here. You have the opportunity for hydrogen bonding here. A little bit of hydrogen bonding with the carbonyl. So those are polar but uncharged R groups. Serine. So that's the second category. Aromatic. Well, let me as I go back here. Let me back up just for a second. You'll see this is one that I want you to memorize. Alanine and glycine. This is the only group where I picked out 2. Glycine because it's unique. It's the only one that's not optically active. It's also the simplest amino acid. Now keep alanine in mind. You can see it has a methyl. It has a methyl right here, right? Let me just go to the here and The one here I think is serine. Pardon me?
Are you saying optically active?
Optically active.
Like the eye, right?
Well, like the eye, it rotates polarized light.
Oh, okay.
That's the optics of it, the optical activity of it. It will rotate polarized light to the left or the right.
All right.
Optically active. No, the one I have chosen here is cysteine in the uncharged group. Now look at phenylalanine. Phenylalanine. So this alanine has a methyl group right here. Well, you take one of the hydrogens off and put a benzene ring on it, a phenyl, and you got phenylalanine. So that's the aromatic one that I picked out. Tyrosine, here's tryptophan. This is the Thanksgiving amino acid, right? Why do I say that?
That's turkey.
Apparently turkey meat has a high concentration of tryptophan in it. So it's a form of protein with high levels of tryptophan, and it makes you feel happy and relaxed, and you go to sleep. And then you add pecan pie to that, and a big mound of mashed potatoes.
Ooh.
And who can stay awake?
Nope.
Nobody. Can't even watch the football game. Who won? You ask 3 hours later. I don't know. Okay.
I'll already say.
Lysine. All right, so here's where you see. Here's the R group. You've got one of these on the R group. 4 carbons and that's lysine, arginine, histidine. All right, so that it's a positively charged R group. You're going to see that the nature of these R groups factor into how these interact with one another and how they help form the protein structure. And then finally, aspartate and glutamate, and I guess I have glutamate here.
Thank you.
Now wait a minute, I thought there was a glutamine. Isn't there a glutamine? Oh yeah. Okay, don't get glutamine mixed up with glutamate. This is an amine, glutamine. All right, so don't get that mixed up with this glutamate. Glutamate doesn't have an amine on it. It has a carboxyl group on it. And if you ever have trouble remembering, refer to the back of chapter 18's homework, because it basically says, draw the structures of these amino acids. And the ones I ask you to draw are the ones I want you to memorize. Okay. Rather than write out the names of these all the time, what we typically do are use these 3-letter abbreviations. So instead of serine, we have SER. Instead of alanine, ALA. And we use those when we're looking at the amino acid sequence in a protein. We will actually lay this out. One other convention is to use these letters for the various amino acids. Some of them are pretty, you can remember. Alanine is A, cysteine is C, glycine is G, but like phenylalanine is what? F. Proline is P, serine. So you can see a whole bunch of them start with the letter T. You can't use the letter T for all of them. You don't have to memorize this, just recognize it and understand what it's trying to tell you. Okay, let's just mention optical activity one more time here. I have a glyceraldehyde floating around somewhere. I thought I did. Oh, there it is. Okay. Remember in chapter 16 on the saccharides, when we introduced introduced the D and L isomers of glucose, for example, we said that glyceraldehyde was our reference molecule. And remember the convention. You put the carbons vertically. You put the carbonyl at the top, and then arrange your carbons vertically away from you, and then pointing out at you is a hydrogen and an OH group on the glyceraldehyde. And if the OH is on the left, it is L-glyceraldehyde. Well, for alanine, we're going to do the same thing. We're using this as our pattern, our reference molecule. We're going to put the carbonyl at the top, the R group at the bottom, and have 3 carbons in a row. 3 carbons in a row like that, fading away from you. And then what's going to be sticking out at you is an amine group and a hydrogen. This and this will be sticking out at you horizontally. And if the amine group is on the left, it's the L. The amine group is on the right, it's the D. And this is the biologically active, the L-alanine. This is what you find in nature, our L amino acids. I remember the first time I saw a bottle of lysine. It said L-lysine. And I go, oh man, this is before I learned. I knew that D-glucose was the biologically active glucose, and an L-alanine. I thought, man, that's the wrong isomer. But no, that's the right one. L-lysine is the right isomer of lysine. And lysine is a essential amino acid. You need to get that in your diet, lysine. So sometimes it is sold as a supplement. I'm not recommending that. Just eat a good diet. Get your lysine from your food. All right, that has to do with the optical activity. All right, now let's start looking at how does one amino acid interact or react with another amino acid. Now here's the beautiful part of this design. You have a positively charged weak base here. So here's a weak base, here's a weak acid. And so the weak acid on one amino acid will react with the weak base on another amino acid, forming water and an amide or peptide link. So this is a peptide bond. But you can see you're removing that water, and you're getting a carbonyl and an NH, carbonyl, NH right next to each other, which is from our organic. This is an amide. In biochemistry, they would just refer to it as a peptide linkage or peptide bond. By doing that, you have left on this end of this dipeptide a nitrogen, and on this end a carboxyl. Okay, what is being depicted in the opposite direction is digestion, or the hydrolysis. The hydrolysis of something like this, adding water to this, so starting with this, adding water, would break it back down into the amino acids. So when you do eat a steak, you're not absorbing protein as protein molecules. You're absorbing protein as individual amino acids. Your body digests the steak or the beef or whatever, digests it and is breaking it down into individual amino acids and absorbing them through the intestines into your body, and then into the cells, and the cells are rebuilding them back into the protein that your body needs. Your protein. What protein molecule do you need? All right? And your body can produce thousands. Every cell of your body can produce thousands of different proteins. All right, so we talk about a dipeptide, a tripeptide, oligopeptides. You saw that prefix in oligosaccharides. 3 to 10 or 11 units long is an oligo. So an oligopeptide would be a short little chain protein, or a polypeptide, 50 or more amino acids. Here is a pentapeptide. All right, now we're starting on the N-terminal end. That's where we start. This is where we start naming the sequence. So what's the very first amino acid? Serine. Right in here is the second one, glycine. Right in here is the third one, tyrosine. Alanine. All right? You can see the methyl right there. That's the R group for the alanine amino acid. And then finally, valine. So really what you have here, if you take a look at this, because your homework is going to ask you to draw the structure of a tetrapeptide. And it's going to give you a sequence like something like this. It's going to give you 4 amino acids, boom, 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 and it's going to say draw that tetrapeptide. So look at what you have here. You've got a nitrogen, you got your alpha carbon, and then you have a carbonyl. And then that pattern repeats itself. And there's going to be a hydrogen on the nitrogen. Hydrogen on the nitrogen. And there's going to be an R group here. There's going to be an R group here. So off of every alpha carbon, go away. There you go. Off of every alpha carbon is going to be an R group. And of course on the end you're going to have an NH3 plus, and on the other end you're going to have a COO minus. This here is laying that pattern out, this slide, just what I just showed you. Let me try to just see what these last couple slides are, and then I'll hand you the Chapter 18 homework. All this slide is saying is, in Chapter 10, early in the semester, I told you that a carbon-carbon single bond had free rotation around it. It was flexible, had a lot of rotation. And I said, when we got to Chapter 11, I said, you get into a double bond and you cannot rotate around the double bond. Remember? Well, what is this slide trying to say? Although this here looks like a carbon-nitrogen bond, and you would expect that you could rotate around it, what it is actually is that this bond here gets shared. We use the term delocalized. It gets shared between this carbon and oxygen and this carbon and nitrogen. So there is no rotation around that carbon-nitrogen bond. It looks like it should, Why does... Well, if you look at oxygen and nitrogen, this does have the higher electronegativity, but that has a pretty good electronegativity too. So even though that has the strongest attraction, that has attraction for electrons too. So they're going to get attracted to the oxygen more, but they get shared with the nitrogen because it's pulling for those oxygens too. You can form carbon-nitrogen double bonds. And so this is a partial one, and this is what it looks like in the polymer. Okay, if you actually look at a model of it, here is a carbon-carbon single bond, rotates around there. Carbon-nitrogen single bond, free rotation, there's all single bonds here. But this nitrogen next to that carbon, because of the availability of these electrons to share between these two atoms, the oxygen and the nitrogen, there's no rotation there. What does that do to the protein? It makes the protein backbone a little stiffer than you might anticipate. It's not like spaghetti that has been boiled long enough. It's like spaghetti that's still kind of stiff. It hasn't really, you only boiled it for a minute or two. And it's got some soft spots, but it's got some hard spots in it. And that's what this has, some firmer areas of it. Okay, that gets us there. That's where I want to pick up. All right. Now here's the way I like you to handle this. I think you can do everything on the back page. You're going to see a lot of references to primary, secondary, tertiary, quaternary structure. You can answer those on Thursday. Turn this in at the end of our class on Thursday. Okay?
On Wednesday.
Wednesday. Sorry. All right? So you can turn it in at the end of class on Wednesday. If you want to look over the front page and make sure you're kind of familiar with it.
Yeah.
That's
I don't know if
not
you
a
can
bad
take
idea.
microbiology at the same time as AP.
When you come in on Wednesday, we'll move the tables around, and I want you to sit no
but
more
I
than
did
3 people. I need 7 tables.
I did AP 1, chemistry 1,
You get one?
and then I did AP 2, chemistry 2, chemistry lab 2.
Okay, see you Wednesday. Don't come on Thursday. I won't be here. If you come on Wednesday, you don't have to come on Thursday.
I'll see you later in the weekend.